Hey everyone, and welcome to this explainer. Today, we're going to explore how computational simulations are completely revolutionizing drug discovery. I want you to think of it as a microscopic dating app for molecules. Imagine trying to find one absolutely perfect match out of millions of candidates, but having to do it at a molecular level. That is exactly what molecular docking does for modern science.
So how do scientists actually find that perfect drug without running endless, exhausting lab tests? Well, let's look at the massive problem with traditional drug discovery. Historically, finding a new drug meant synthesizing and testing thousands of compounds in a physical lab. We are talking about a process that takes years and millions upon millions of dollars. But what if a computer could just do all that heavy lifting first? Computational shortcuts, like molecular docking, provide this incredibly powerful solution. They give researchers a way to basically peek at molecular interactions before a single test tube is ever used.
Here is our roadmap for the explainer today. First, what is molecular docking? Second, the core biological components. Third, types of docking simulations. Fourth, the 5-step docking workflow. Fifth, interactions and docking scores. And finally, real-world docking applications. Let's get into it.
Section 1. What is molecular docking? The computational shortcut. At its core, molecular docking is a computer-based simulation that predicts the preferred orientation of a molecule when it forms a stable complex with another. You see, it's not just about whether they fit together, it's about exactly how they fit. The software predicts the best binding position, which we often call the pose, as well as the orientation and the affinity, or the interaction strength. This is a massive deal because it saves unbelievable amounts of time and money, helping researchers prioritize only the absolute most promising candidates before heading into the lab.
Section 2. The core biological components. The matchmaking clients. To really get this, let's meet the clients in our microscopic matchmaking scenario. First up is the receptor. Think of the receptor as a highly specific lock waiting for the perfect partner. Biologically speaking, this is a large macromolecule. It could be an enzyme, a cell surface receptor, a viral protein, or a bacterial protein. Basically, it's the large, complex target that we are trying to influence.
On the flip side, we have the ligand. If the receptor is the lock, the ligand is the key trying to find the perfect fit. This is typically a much smaller molecule. We're talking about a synthesized drug molecule, a peptide, a small organic molecule, or even a naturally occurring compound. The ultimate goal here is to figure out if this specific ligand can successfully interact with our receptor. And of course, they need a place to meet. That exact meeting place where our two clients connect is called the binding site, or the active site. Now, their compatibility is entirely determined by their unique shape and chemistry. A ligand can't just slap onto the protein anywhere. It has to fit perfectly into this highly specific region, much like a key sliding right into the precise grooves of a lock.
Section 3. Types of docking simulations. Setting the rules of attraction.
Just like any matching algorithm, we have to set some rules. Bioinformaticians generally take one of 3 distinct approaches here. With rigid docking, both the protein and the ligand remain completely fixed in shape. It's really fast, sure, but it's less realistic because biology is inherently dynamic. On the total opposite end of the spectrum is flexible docking, where both molecules can change their shape. This produces highly accurate predictions, but it eats up massive computational resources. So right in the sweet spot, we have semi-flexible docking. The protein lock remains fixed, but the ligand key is allowed to shift and adapt its shape. This is the most commonly used approach today, simply because it strikes that perfect balance of accuracy and efficiency.
Section 4. The 5-step docking workflow. The algorithmic pairing.
So how does this actually happen? It's a very methodical 5-step journey. Step 1 is target selection, where you usually download a 3D protein structure straight from the protein databank and pinpoint that active site. Step 2 is protein preparation. You can't just use the raw file. You have to remove unnecessary water molecules, correct missing residues, and add hydrogen atoms to optimize the whole structure. Step 3 is ligand preparation, which means optimizing its geometry and assigning atomic charges so the computer actually understands its chemical nature. Then we hit Step 4, the docking simulation itself, where the software generates multiple binding poses and scores them. Finally, Step 5 is result analysis. This is where researchers evaluate the binding energy and look really closely at the chemical interactions.
Section 5. Interactions and docking scores. Rating the match.
Once the software introduces our molecules, we have to see if they actually have chemistry together. There are 5 specific types of biological forces that hold our lock and key in place. Hydrogen bonds are absolutely crucial for highly specific, stable connections. Hydrophobic interactions bump up stability by minimizing the molecule's contact with water. Then you have electrostatic interactions, which happen between positively and negatively charged groups. We also look at van der Waals forces. These are weaker attractions, but they still significantly contribute to the overall bind. And finally, pi-pi interactions occur between aromatic rings and play a massive role in a lot of drug-protein interactions. The more of these harmonious connections the software finds, the better the match.
All of those interactions boil down to one crucial metric, the docking score. This score ranks thousands of compounds to tell researchers exactly which matchmaking attempt was the most successful. When a computer runs a high-throughput screening of a massive library of compounds, it uses this score to estimate the binding affinity. It literally generates a ranked list, placing the most promising drug candidates right at the very top for further review.
Now, when you look at a docking score, you're going to see a number like negative 9.5 kilocalories per mole. And this is where it gets a little counterintuitive. A more negative binding energy actually means a stronger, more stable predicted interaction. A less negative, or even a positive number, indicates a much weaker prediction. So, in the world of molecular docking, researchers are constantly hunting for the lowest, most negative numbers they can possibly find.
Section 6. Real-world docking applications. The successful marriages.
Let's talk about the heavy-hitting software programs that act as the actual matchmakers here. Autodock is incredibly popular, largely because it's a powerful and free tool. Autodock Vena builds on that legacy with much faster calculations and improved accuracy. Now, for commercial labs that need elite precision, Schrödinger Glide is a major go-to for high-accuracy scoring. Gold is super fascinating because it actually uses genetic algorithms to handle flexible docking. And we can't forget tools like USCF Chimera, which are absolutely vital because they allow scientists to visually analyze these complex molecular interactions in 3D space.
The real-world value of this technology is just immense. It's driving fast-track solutions across so many vital fields. In cancer research, scientists use it to discover inhibitors for cancer-related proteins, which pushes us ever closer to truly personalized therapies. During infectious disease outbreaks, it rapidly identifies potential antiviral or antibacterial compounds. It's even heavily relied upon in biotechnology for enzyme engineering and in natural product research to screen plant-derived phytochemicals. It's basically a way to check if nature has already built the perfect medicine for us. But, as with all scientific tools, we have to keep ourselves grounded and look at the full picture. Yes, molecular docking saves incredible amounts of time and reduces laboratory workload while supporting rational, high-throughput drug design. However, it definitely has limitations. For one, these predictions are entirely dependent on the quality of the initial protein structure you feed into the system. Plus, protein flexibility is sometimes really hard to fully represent, and complex solvent effects, like exactly how water behaves around these molecules, are often simplified by the software. Because of all this, we have to remember it's still just a prediction. It ultimately requires strict experimental validation in a real physical lab to confirm the biological activity.
Which brings us to a really exciting question. As we look to a future of cloud-based virtual screening, vastly improved scoring algorithms, and personalized genomics fully integrating with AI and machine learning, how long will it be until computational design perfectly replaces the test tube? Will there actually come a day when our digital molecular matchmakers become so flawless that we can take a drug straight from a computer screen and give it to a patient? It is a fascinating frontier in bioinformatics, and it's advancing faster than ever before. Thank you so much for joining me for this explainer, and keep exploring.